Marshall. Dr. Marshall is a clinical laboratory director in the Division of Genomic Diagnostics at the Hospital for Sick Children and assistant professor, laboratory medicine and pathology at the University of Toronto. With expertise in clinical diagnostics and translational genomics, Dr. Marshall's research focus is on applying the latest genome sequencing technologies to end the diagnostic odyssey of patients living with rare disease. Please join me in welcoming Dr. Christian Marshall.
Well, thank you very, very much for the introduction and for the invitation to speak here. I'm going to talk a lot about genome research and diagnostics in Canada, and specifically, how we use research discoveries and translate those into diagnostics and healthcare. I'm going to frame my talk around this concept of translational genomics. I spend a lot of time, most of my time in the diagnostic lab, so I'm really interested in how we make or figure out these research discoveries, and as I said, translate them into healthcare. As Dr. Green said, I think ultimately any research discoveries should really lead to the improved patient health. So we have these genome centers. They sequence a lot of data, sequencing at scale, for research discovery and technology development. That data then can be, or that technology development and expertise discovery can be translated into hospital-based diagnostics. Specifically speaking, I do a lot of work on rare disorders in a pediatric hospital. I think that's where it has a lot of impact right now, but as Dr. Green said, ultimately there will be a lot more impact for more common disorders that occur later in life. We do sequence generation for diagnosing of patients. We are technology, we adopt the technologies that are sort of developed in the research world. And what I'm going to try to convince you a little bit is that we are now playing a role in being able to get a lot of that data back into the research world. So there's a lot of data that is being generated, a lot of genomics data that's being generated as part of clinical care for those cases that remain unsolved. We can move that back into research and hopefully continue this cycle. I'm going to talk a little bit about these genome centers. Canada's National Platform for Genome Sequencing, or CGEN. It's an integrated national platform with 3 different nodes in Toronto, Montreal, and Vancouver. This has the large-scale sequencing infrastructure that's needed to support a lot of these projects, including both short and long-read sequencing. You can see from the map there's roughly over 1,100 research groups as users, and you can see that distribution across Canada. Since 2015, this entity has sequenced more than 140,000 genomes. Not all of these are human, but about 65% of them are. You can see the cumulative genome sequence there. Last year, just alone, about 16,000 human genomes were sequenced. And just to kind of put this in context, I kind of like this analogy. This would be around 30,000, the data anyway would be somewhere around 30,000 4K movies. It would take a long time to watch. So with all these large-scale, or with this infrastructure, you can do a lot of interesting projects. I think I'm going to talk about 2 of them that I've been peripherally involved with. One of them is uncovering the genomic contributions to autism. So this is led by Steve Scher. It's been in this database called Missing, or the project is called Missing. Its goal is really to provide the best resources to enable identification of the subtypes of autism, which may lead to better diagnostics as well as personalized or more accurate treatments. Roughly about 13,000 genomic sequences are available in this database. About 6,000 of those individuals are diagnosed with autism, and the rest would be their parents. This is a nice summary paper from Brent Trost that was published in 2022, just kind of uncovering the genomic contributions to autism. And as I said, we use a lot of this information now in diagnostics. So if you were to sequence the genome of an individual with autism, around 15% of them would have a rare variant that is probably contributing to the diagnosis. Considering the genomic architecture, it's really interesting in that a lot of these variants are sequence-based, but a lot of them are structural variants as well. So a large number of them and a smaller number are mitochondrial variants. I think what's also very interesting is that genomes are actually needed to provide the full picture of the variant contribution to autism. Actually having all the data and looking at all the different types of variation. As I said, this data can then be used to inform diagnostics. Another project that I'd like to highlight is the Care for Rare Consortium for Rare Disease. This is a Canadian consortium that's it was set up over a decade ago to improve diagnosis and treatment of rare disorders. And we heard a little bit about that from Dr. Green. This is 21 sites, 200 physicians, and 100 scientists across the country, so it's really pan-Canadian. Around 5,000 patients and families have been recruited worldwide. This has led to the discovery of 157 novel disease genes or mechanisms. And even more importantly, about 1,000 different patients have been provided with a diagnosis as you move this into clinical care. This is an interesting summary that was published by Kim Boycott in 2022 in the American Journal. I wanted to highlight how gene discovery is now being done. It was really driven in the beginning by, it was sort of at the early days, the low-hanging fruit, looking at just specific cohorts. So this is sort of in that orange color. And then as you move into about 2021 here, a lot of the, sort of more common rare disorders have been already discovered. And so a lot of gene discovery is done now predominantly by data sharing. So that's all that sort of blue there. So you can see in 2021, most of the gene discoveries were because of data sharing. And this is going to be a concept that I'll talk about a little bit later as well. So try to think about these large-scale genome projects and how do we get this into healthcare? Well, this is a map of the clinical genomic sequencing in Canada in 2020. So even though short-read genome sequencing technologies were really mature at that time, we weren't using them in Canada to their full capacity. So clinical cases were undergoing exome sequencing. This is all out of country, so it might be surprising for you to know that there was no lab in Canada that was performing clinical, even exome sequencing in 2020. You can see the map there. Those are the different populations. And underneath, cumulatively, I'm showing about 1,300 families would have been sent to the U.S., mainly to the commercial companies to have exome or genome sequencing. And the other thing you can see is across Canada, it's very disparate. Provincial healthcare is under provincial jurisdiction, and often it's disparate across the country. We sort of looked at this and said, one thing that we wanted to do was kind of repatriate these tests back into Canada because a lot of that valuable data was often lost and siloed. So we thought, can we implement this can we implement genome sequencing in clinical care in Canada, in the province of Ontario? A lot of barriers to this, including thinking about setting up the capital equipment. The data management is one thing that is particularly tricky. Lots of lack of standards, and the test complexity. And so we were engaging the Ontario Ministry of Health at the time to be able to figure out how to do this, because we knew from these large-scale genome projects that this was a valuable thing to do. As I said, we started engaging in 2015, so really early on when we knew that genome-wide sequencing of individuals with rare disease was going to be important in healthcare. This was a collaboration between us at SickKids and also CHIO. And the Ministry of Health, who is the public payer. And so out of this came a platform called Genome-Wide Sequencing Ontario. This platform was launched in 2021. And the idea was to repatriate the test from all the data that was going out of country. We were doing sequencing of exomes and genomes at the time using a hub and spoke model. The reason why we were doing exomes, which is essentially a slice of a genome, was mainly for cost considerations. But as costs continue to come down, we're switching more and more to full genome sequencing. The important thing here was also collecting data to assess the outcomes and inform future adoption. So this, I think, was really important. We were able to collect a lot of the data as we went along to be able to say, what's working and what's not. And as I said, we are using this hub and spoke model where all the DNA comes to SickKids, and we do the sequencing here, and then it's a distributed reporting model. This is a summary of where we're at now. In terms of after running the program for about 3.5 years. About 3,000 families have been reported. We carefully look at their applications. The clinical criteria for actually accessing this test is relatively strict at the moment, so we have to sort of be the gatekeeper. This is going to broaden over time, but about 96% of the applications were approved. We've now sequenced about 7,200 individuals from about 3,000 families. Just under a quarter of those are genomes at this time. Our turnaround time is roughly averaging about 12 weeks, but 87% of the time we're under 12 weeks. And importantly, about 29% of the individuals actually receive a diagnosis that can then help inform patient management. We're continuing to increase and scale up production for this, and we're working with the Provincial Genetics Program to help inform policy. And I wanted to sort of switch gears here and talk about the observations and current directions from having run this program for 3.5 years. So the first observation is the diagnostic yield is relatively high. 29–30% is actually pretty good for a genetic test. And we really think that this is pointing to an unmet need. We need to study the yield of broadening test eligibility criteria and inserting essentially the genome in an earlier part in the clinical pathway. Often these tests are actually done at the very end of a diagnostic odyssey. We're really thinking about that genome first approach. The other thing is that a lot of these cases, despite having a diagnosis, many of them have inconclusive results. So they have sort of these variants of uncertain significance. We don't know what they do yet. Or in fact, they're completely negative. There's 40% of cases have no variants that are reported that we can see have any relation to the reason why they were referred. So we're really thinking about leveraging existing and new approaches to resolve this uncertainty and solve cases, and I think this is where that interaction between the genome centers and research is really important with the diagnostic lab. And lastly, and most importantly in my mind, is that we're accumulating a lot of data in the clinical world. And I think unlocking it and getting it back into research is going to be extremely important. So those data sharing opportunities to aid in that interpretation, build cohorts, understand, to study disease and natural history. The interesting thing that we've done now is because we have a genomic platform that is clinically validated, we can participate in a lot of these sort of clinical research studies. And I'm going to talk about one now. So this is sort of this first thing, broadening the eligibility criteria for rapid sequencing. So this is a project that's part of an international precision child health partnership that is between Sick Kids, Boston Children's, Great Ormond Street in London, and Murdoch Children's Research Institute in Melbourne. And one of the flagship projects we have here is called Gene Steps. And this is, what we're trying to do here is perform rapid genome sequencing on newborns presenting with seizures with the aim to look at their diagnostic and clinical utility. Right now, these individuals presenting with just seizures, a single system sort of disorder, they're not eligible for our actual GSO platform, and we're trying to develop the evidence to show that it should be. So because we have that infrastructure in genome diagnostics, we can go clinical genome sequencing on these individuals and then study the outcomes. The first paper that we published, you can see in Lancet Neurology, showed a diagnostic yield of 43%. And importantly, a median time of seizure onset to test results of 37 days. That might seem like a long time, but it's actually pretty good. And this is really that sort of precision medicine that we're talking about. Just as an update, we've now reported 108 different families. The yield is about 44%. These families also undergo standard of care testing, which is panel testing, and approximately a third of those remain, a third of what we're finding you can't find on a panel because we're taking obviously a broader approach. This is a really good case example to bring this point home. This is a male that presented at 4 months with generalized seizures and was started on that first-line treatment. We were able to do the rapid genome sequencing. We were able to report 2 days, actually was reported 2 days before he represented to the hospital with the recurrence of seizures. We found a pathogenic variant in a gene known to cause seizures in PRRT2. This informs management. We can then change the treatment, immediately started on a different medication, and has been seizure-free since. So this allows for that sort of discontinuation of unnecessary treatments, avoidance of additional investigations. And in terms of genetic counseling, this individual is now expected to have normal developmental trajectory. And so this is really that personalized medicine approach and being able to use the genomic information in a way. And we're just trying to do this now faster and faster and faster. Another observation is obviously new approaches to resolve uncertainty and find new diagnosis. So I'd just like to go through a quick case study here. This is a 7-year-old male with developmental delay and hearing loss. They had a lot of previous genetic testing, and I think this is a theme. This is a classic diagnostic odyssey where they keep undergoing different tests that keep being negative. They had clinical exome sequencing quite some time ago. We enrolled this patient into genome, into a research study using genomes at the time. We uncovered 2 different variants in this gene called ISEC1. There was a paper published relatively recently showing that biallelic variants, so you need to have 1 on each allele. This is a recessive disorder, cause this intellectual disability. The interesting thing that we found here was that we had 1 variant that was actually inherited from the father. The other variant was de novo, and so we didn't know where it came from. And so we couldn't actually confirm a diagnosis in this case because we didn't know if it was recessive or not. And so short read sequencing has a ton of it, has been, obviously the sequencing technology that we're using mostly, but it also has some flaws, whereas long read sequencing has some advantages over short read sequencing, and this is sort of what we're studying now in the research world. So you can see here there's a gene, there's a short read sequences, and the long read sequences are what they sound like. It's just the longer stretches of DNA that then you can easily map easier back to the genome. And this has advantages in improving the detection of other types of variation, like structural variation. You can sequence through repetitive regions of the genome that short reads sometimes can't map to. We can do phasing, which we're going to talk about here, and an improved discrimination of pseudogenes. And the 5th one that Dr. Green also mentioned was it actually is sort of more real-time sequencing and sequencing of the native bases of DNA. You can actually see methylation modifications on the bases as well. So we did this for this case in the research context and found out that this is just a diagram showing a genome alignment. You can see the variant itself was actually found to be phased onto the paternal haplotype. So what this means is that the de novo variant occurred on the paternal haplotype. It means that both variants are on the paternal haplotype, and essentially we were able to use long read sequencing here to provide evidence to rule out this gene. So we ruled that out, and then we can actually use the data to search for another candidate. And that's what we did. Long read sequencing revealed a new candidate, and so this again is another sequence alignment. And in that red box, you can see a deletion that we found overlapping this gene called DYRK1A. So this was a 2 kb de novo deletion. Arguably, you might be able to find this on short read sequencing, but it could be potentially tough. Long read sequencing was able to find it. This is associated with an intellectual developmental disorder, which sort of matches the phenotype quite well. There's phenotypic overlap with our program, but really we don't really know the effect of this deletion. It overlaps 1 exon. It's hard to say exactly what it's doing in terms of changing the RNA. We could do RNA sequencing, but what's interesting about this particular gene is it does have a methylation signature. And so we were able to actually use the genomic information to actually do methylation analysis all in one experiment. So this is work that we've been doing with Rosanna Wexberg's lab. And essentially what this is a plot, a PCA plot with clustering based on the methylation analysis. And you can see on the sort of the one side, the ones in red and in purple are sort of controls, and you can see our sample in gray is the one that is sort of an outlier. And so it's an outlier, it doesn't really cluster with the rest of the cases. And so this potentially has a methylation effect, or this variant could have a methylation effect and maybe more of a gain of function type variant. It doesn't actually fit with the normal epigenetic signature of variants in this gene, but it may have a different effect. So long read sequencing in this case can sort of help determine pathomechanisms in some cases. And so I think we're getting to a point where potentially long term, we will have an all-in-one sort of genomic test that will provide us with even more information than what we're seeing now. And the third thing I wanted to highlight was data sharing opportunities at a national scale. And so I talked a little bit about the different labs across the country and all trying to repatriate genomic testing. These are these different funded projects from Genome Canada. They were called GAP grants, and this was sort of really trying to get the clinical sequencing up and running And as I said, like this was what sort of funded our project for the GSO. But you can see the other different provinces have been doing the same thing. And if you sort of estimate this, there's about 10,000 clinical grade genome-wide sequences from about 5,000 different rare disease probands that can be used to help further interpret data. And over top of this, obviously, and Dr. Green talked about a lot of this, like some of the, what are the ethics and policy around this? You need to have this policy toolkit. But we were thinking about this concept of a health data ecosystem. And the health data ecosystem is really a way to figure out how to share a lot of this data to be able to interpret new data, and also to be able to send a lot of it into research. About 90% of the patients that come into the clinic, about 90% of them opt in to sharing of their genomic data that's done on a clinical research, or on a clinical basis. And so we can do interesting things. And one thing that we have right now is a sharing agreement between us and the Children's Hospital of Eastern Ontario. And so this is just an example of, once we've sequenced 3,000 individuals, we can really get a sense of what is the landscape of rare disease and diagnosis in Ontario. So out of about almost 800 diagnostic cases, we have a lot of different loci because these are rare disorders, but many of them are repeat diagnosis. And you can see, it's a little hard to see, but you can see the top one is Kabuki syndrome. So we have 16 cases that have been diagnosed with this. And why is this important? Well, it's important to have all the data in one spot. You can actually start building cohorts for further study, identify candidates for clinical trials, study natural history, get case series together, et cetera. And our vision is to really do this on a national level. And so I'll sort of finish by talking about this vision of the health data ecosystem in Canada, where really we're thinking about the patient journey should involve the ability of individuals that are undergoing clinical testing to be able to be involved in research. This is going to be complicated, but it's something I think we can do. This is a rare disease family. They go and do their diagnostic tests. We have all this in blue. This is the hospital where we hold the data, but then consenting for the release of that data into sort of more of a research arm to be able to actually do more research, be open to researchers. We can make more discoveries with the data and then feed that information sort of back into the diagnostic lab. So here's a summary on future directions. I've told you a little bit about the Canadian Genomics Enterprise. It's providing that national infrastructure to be able to do research on a large scale. Where findings can be translated into diagnostics. We've got many challenges to the implementation of clinical genome sequencing, including a lot of the technical, financial, and healthcare education, but it's now part of routine service, so we're actually doing this now. Genomic data generated for clinical purposes can further be released into that research to drive new discoveries, and especially using the latest technology. So I think there's a lot more interplay now between the genome centers and development, and trying to translate that information back into diagnostics. And certainly driven by that decreased cost, a large proportion of the genomes we think sequence will be carried out as part of routine healthcare, and we really need to develop a lot of these strategies like a health data ecosystem need to be developed to share this data and make it accessible. With that, I'll end. I'm happy to take questions. I'll just leave the acknowledgements up. This is speaking on behalf of a lot of different people, both at Sick Kids Hospital, but across the country, in the Care for Rare group, and the Gene Steps team. Thanks.
Thank you very much, Dr. Christian Marshall. That was a great talk. We have a question already from the audience. First one is, I know you mentioned a lot about genomics, but can you discuss the role of RNA sequencing also in enhancing diagnosis?
It's a great question, and also something that I had a bunch of slides on, but I didn't have time to talk about. So with an RNA-based, using the same sequencing technologies that we're talking about, we've now sort of validated that in the clinical lab. I think a lot of these variants that are coming up that are of uncertain significance, especially non-coding variation that may be involved in splicing, you need the RNA to be able to figure that out. Right. And figure out what the effect is on that transcript that's leading to effect on the protein. And so from our own data, we found that about 5% of cases in our clinical cohort have variants of uncertain significance that RNA sequencing would be beneficial. It's not covered right now as part of clinical sequencing, but it's something that we are trying to get sort of funded.
Okay. And the next question is, I think we appreciate the bigger sample sizes whenever we can. And in that case, have you networked with US and Europe for increasing the sample size and data?
Yeah, so that's another great question. I mean, the short answer is yes, in the sense that, there's public databases that are available that we can use. We have lots of collaborators in the US. I think the challenge is a lot of these sort of agreements, our data is stored in a certain cloud environment that is not easy to sort of, go back and forth between, different cloud instances that are sovereign. So all of our data needs to be stored in Canada. And so that makes it a little bit more difficult to do this, at least at a huge scale. But I think it's something that we need to tackle.
And I think along that line of data, I think you're absolutely correct, because different facilities use different machines and different results in different data sets. What are the barriers and how can you standardize our processes moving forward?
Yeah, so that's something that is sort of, been something that we have struggled with a long time. I think the technology is moving really, really quickly. And, the algorithms are moving really quickly. So there's this, thinking that you have to have everything exactly the same in terms of, how the data is generated and then sort of reanalyzing it, at least to be able to have everything that is, completely standardized. But I think it's a massive challenge. Going back and reanalyzing data is a huge task, especially if you're trying to store the genomic data for a long time. It can be a monumental one. So I think there's other sort of clever ways of trying to put data sets together, or at least understand, at least on whole, what they look like, and then sort of making comparisons in that way. But it's very difficult to sort of standardize across everything. The technology moves too quickly.
Okay. And we do have another question. It says, what are your thoughts on having exome sequencing standard at birth for all?
My thoughts on exome sequencing is that we need to move away from it, to be honest. We should be doing genome sequencing. I think now with the latest versions of the sequencing technology and short-read sequencing technology, I think we're finally there, at least in the clinical lab, to be able to move everything to genomes. So that's our plan within the next few months. Everything will be genome sequencing. And I foresee a day when individuals are coming into the hospital, their genomes are being sequenced, and it's just a matter of, are we looking at a single system or are we looking genome-wide? But the backbone will be the same test.
Okay. And you mentioned about short read versus long read, and we saw a clear benefit of the long reads in this case. What prevents or what are some limits and barriers in using long read all the time in this scenario?
It's the age-old issue of cost and throughput, right? So right now, because the short-read sequencing technologies have advanced so quickly, we've been able to increase throughput. That has decreased cost. We can sequence 128 genomes every 2 days in our lab. You could sequence maybe 16 long-read genomes in the same amount of time. And so we just don't have the capacity, and it's roughly about 4 times the cost at the moment. So I think those are the biggest barriers. This will probably change over time, just like everything. Long-read sequencing is where we were with short-read sequencing 10 years ago, at least in what I've seen.
Okay, perfect. Thank you very much.